You are once again welcome to Bio 211B Plant Physiology, Unit 6, Session 5, Determination of Transpiration. And by the close of this session, students should be able to determine transpiration in whole plants or leaves. Compare methods of determining transpiration in plants and describe some methods used in measurement of transpiration in plants. Transpiration can be measured using various methods, and these include the weighing method, cobalt chloride technique, bell jar method, photometer method, and by using a porometer. However, this does not prevent you from learning to use any other method not described here. The cobalt chloride method. This procedure is a simple one for the determination of transpiration since it requires no elaborate equipment and determinations can be made simply and quickly. The equipment and material required are a leafy twig or a small plant standing in water or a potted plant, sellotape, blade or a pair of scissors, cobalt chloride paper, and the method is as follows. The first point is that you obtain two small pieces of cobalt chloride paper. Cobalt chloride paper looks blue and turns pink when moist. This change in color allows us to find out if a plant is transpiring or not. And secondly, in the laboratory, use a small leafy twig of a tree or shrub and let it stand in water. Otherwise, you can use a potted plant. One can also use the leaves of any plant in the schoolyard or garden or at home. However, a potted plant will be handy and excellent for the experiment. With the sellotape, stick one piece of cobalt chloride paper to the upper side of the chosen leaf. You must completely cover the piece of cobalt chloride with sellotape to ensure that any moisture on the cobalt chloride comes from transpiration from the leaf. Now stick the other piece of cobalt chloride paper to the lower side of a different leaf the same way as in step 3. Make sure that the main veins are avoided to enable the sellotape stick well and tightly onto the leaf surface without leaving any gaps for external moisture to reach the cobalt chloride paper. You are then ready to find out if transpiration takes place and how fast for each leaf surface. Hence, you must note the time and keep observing the two pieces of cobalt chloride sellotaped to the leaves at regular time intervals. For example, every 5 seconds. The observations are that after some time, you would observe that the cobalt chloride has changed color from blue to pink if transpiration occurs in the leaf. If you answer the following questions, you should be able to tell how fast transpiration occurs in the lower and upper surfaces of the leaf. The questions are: 1. How long does it take for the first trace of pink color to appear on each piece of the cobalt chloride paper? How long does it take for each piece of cobalt chloride paper to turn completely pink? And does the upper or lower surface of the leaf lose water faster? Why does one side lose water faster than the other? So the photometer method. With the aid of a photometer, the effect of various environmental factors on the rate of transpiration can be studied. It must be noted that there are other types of photometers which are not described here. As water is transpired or used by the plant to replace transpired air, it is replaced from the reservoir shown. This pushes the air bubble to the left, providing a precise measure of the volume of water used, and this can be readily seen in your textbook. It is a simple method used to demonstrate how the rate of transpiration in plants, particularly using twigs or young plants. This method can also be combined with the determination of density or stomata per leaf area in different plant species to determine the effect of differences in stomatal density of rate of transpiration. The procedure is outlined as follows. A sharp blade is used to cut a leafy shoot underwater. The leafy shoot is then inserted through the hole of the stopper provided with the potometer. The potometer is filled with water and the stopper holding the leafy shoot fitted to the apparatus as displayed in your textbook. So Vaseline or any grease is used to seal all the connections of the apparatus to prevent air from entering. If air enters, it will cause a situation like cavitation. However, since the apparatus is artificial, another channel for water movement cannot be created. This will affect water uptake. Air bubble in the capillary tube is trapped by doing the following. Dip the end of the capillary tube into the beaker of water. Close the tap reservoir. Take away the beaker of water and allow the plant to transpire for a while and re-immerse the capillary tube into the beaker of water again. After this, wait for about 5 minutes for the plant to equilibrate. The rate of transpiration is then estimated by measuring the distance moved by the air bubble per unit time. This means that you need a stop clock to check rate of transpiration with time. Another measurement or reading is taken and the average of the two readings computed as the rate of transpiration. Now determine the total leaf surface area of the experimental plant using a graph paper. Transpiration rate can be expressed in terms of water transpired per unit time per unit area of leaf surface. The experiment afterwards, the volume of water transpired can be estimated using the formula distance moved by air bubble, that's D in centimeters, cross-section area of capillary tube, A equals cm squared. Therefore, volume of water transpired, V equals D by A cm cubed. This experiment can be carried out under different environmental conditions, such as under direct high light illumination using a bulb, under low light condition, in rapid air movement, which can be done using a fan with different speeds or in still air. You should know that in measuring the total surface area of leaf, both the upper and lower surfaces should be included. Also, note that the potometer only measures the rate of water uptake, hence it cannot be used to find out the actual transpiration rate. The gravimetric method. One can also measure the rate of water loss in a leaf, twig, or a young plant using the gravimetric method. This is also referred to as the weighing method. The materials and equipment you require as follows: a leafy shoot or twig or a potted plant, a measuring cylinder, an electronic balance, polythene bag of a potted plant is used. One may also need a retort stand for the twig or leafy shoot experiment, a rubber band. The methodology. Here we shall discuss the experiment using the potted plant. Obtain a potted plant and a large polythene bag. Make sure the plant is well watered to fill capacity. Allow the excess water to drain and the soil sufficiently moist. Now place the potted plant in the polythene bag. Secure the top ends of the polythene bag as it ends round the base of the potted plant. That is the essence of doing this. Write down your answer before continuing. This is to prevent water from evaporating from the soil surface so that we can determine the actual rate of water loss by the plant. Now place the plant in the polythene bag on the electronic balance and note the initial weight. Leave the setup in the open and record the weight every 30 minutes for the next 3 hours. Now answer the following question. What is the amount of water loss for each time interval? What is the rate of water loss for each time interval? What is the amount of water loss over the duration of the experiment? What is the rate of water loss over the duration? With this set up, you can find out the rate of transpiration under various environmental conditions. For wind, one can use a fan. For light intensity, different bulbs of varying wattage can be used. How would you determine the rate of transpiration at different temperatures using this method? Find out how you would use a twig or a leafy shoot to determine the rate of transpiration. So in review, three methods were discussed. One, the cobalt chloride paper was outlined to show the transpiration occurs in leaves. And two, the porometer, the photometer, and the gravimetric method were discussed to show how they are used to determine the rate of transpiration.